Hello, my name is Patricia, and today I'm going to talk about my project on altered expression of RAG-GFP in not aim to knockout mice in their primary and secondary lymphoid organs. So type 1 diabetes is a chronic inflammation driven by immune factors that cause destruction of pancreatic beta cells within the islets of Langerhans. Non-mice, non-obese diabetic mice are used as spontaneous model to study this disease because they exhibit similar immune defects that are also observed in human type 1 diabetes patients. Although there have been research done on this and numerous factors contribute to the onset of type 1 diabetes, the exact underlying cause of this autoimmune disease has yet to be determined. Understanding the pathway and molecular interaction are essential for development of therapies for prevention or even the reversal of this autoimmune disease. Previous research done in the Tisch lab has shown that non-mice deficient of inflammasome AIM2 molecule, also referred as AIM2 knockout mice, have shown reduced type 1 diabetes incidence. We know that autoreactive T cells are the primary mediators of this destruction. Therefore, we suggest that there has been altered T cell development in AIM2 knockout mice that contribute to the delayed onset of type 1 diabetes. T cell development in the thymus are dependent on TCR rearrangements. Also, we know that during negative selection, those highly autoreactive T cell needs to be deleted. And we know that RAG enzyme signals for TCR and BCR rearrangements. Meanwhile, CD5 expression is tightly correlates with TCR signal as well as affinity for self-peptide. Therefore, our project focused on RAG expression as well as CD5 expression. To do so, we generate AIM2 knockout RAG-GFP knock-in mice to test our hypothesis that deficient of AIM2 molecule cause altered T cell development and output that results in maybe potential reduced beta cell autoimmunity. These mice are generated by the Tisch lab. And first, we use a TAL PCR to confirm its AIM2 genotype. On the left lane, you have the AIM2 knockout mice, and on the right lane, you have the not wild type with a heavier lane. Then we use flow cytometry to confirm its RAG-GFP expression using their blood periphery lymphocytes. So mouse A is a RAG-GFP negative mice and mouse B is a RAG-GFP positive mice. Then we use thymus and spleen single cell suspension first stain with a live dead discriminator dye, then stain with other extracellular antibodies and fix with PD fixed perm. And then we analyze using an LSR2 instrument and analyze using flow gel. Let's take a look at results. We'll focus on thymus for now. We find that AIM2 knockouts show a similar frequency of thymocyte subsets, the double negative, double positive, CD4, CD8 single positive cells. This suggests that AIM2 knockout might show no inherited difference in T cell development comparing to the not wild type. However, we do find there has been elevated RAG-GFP expression in thymocyte after double positive stage in these three populations. And such difference is maintained when they are egressed into the spleen. As shown here, we have elevated CD4, CD8 percentage of RAG-GFP positive cells. And this is suggesting that we might have increased thymic output in AIM2 knockout mice. However, we do not observe any difference in percent of splenic T cell. Why is it that? We then find that there has been increased percentage of RAG-GFP positive CD11C cells. These are dendritic cells that engulf dying T cells. So the increased percentage of RAG-GFP expression in AIM2 knockout mice suggests a decreased T cell viability, and such decreased viability might offset the thymic output we mentioned above and results in this scenario. Then we look more into the RAG-GFP expression. And evident here, we see that the increased RAG-GFP expression in AIM2 knockout mice are supported by intermediate and high population in CD4 and CD8. Although it's not our primary goal to look at B cell, but we do see that there's no increased percentage of RAG-GFP expression in B cell However, we do see an overall increase of RAG-GFP expression in CD4, CD8, and B cells. Such uniform increase in T cell as well as B cell suggests that the increased RAG expression we see is not due to thymic output, but dysregulation of RAG. We know that RAG is suppressed by TCR signaling. And this can be shown also here, that we see that after double positive stage, there's been a decrease of RAG expression. So the increase of aim to knockout RAG expression suggests that we have reduced suppression. In other words, the TCR signaling in aim to knockout mice might be insufficient to downregulate RAG expression. To confirm that analysis, we study more on CD5 expression. So CD5 expression is directly proportional its TCR signaling as well as affinity for self-peptide. And we see that in aim to knockout mice, there's decreased CD5 expression, suggesting that the TCR signaling is diminishing in aim to knockout mice. And the increased CD5 expression in the single positive showed that there has been increased affinity for self-peptide, so potentially more autoreactive T cell exiting in NOD mice. Looking through literature, we have found that AIM2 molecule actually interacts with other downstream signaling molecules, such as the AKT pathway, to downregulate RAG expression. However, its role in T cell, B cell was unknown. Further studies are required to either directly look at AIM2 molecule interaction with other signaling pathway, or look at AIM2 knockout mice, how the TCR signaling events are dysregulated in these mice. To do so, we have suggested that we can upregulate TCR downstream molecules such as Neur77 by generating a NOD and AIM2 knockout Neur77 GFP reporter mice. Either through direct stimulation or activation, we can study downstream TCR signaling events. Overall, our research has shown that the reduced incidence of type 1 diabetes in AIM2 knockout mice is not due to difference in T cell development or T cell output, but rather distinct TCR signaling events and also decreased self-peptide sensitivity. Thank you.